Hello everyone, welcome back. In this video, we are going to study the notes of first PUC Biology, Mineral Nutrition textbook question answers. First question, methods to study the mineral requirements of plants. Julius Van Sash 1860 demonstrated that plants could be grown to maturity in a defined nutrient solution and in the complete absence of soil. Hydroponics has the following uses. The essential elements have been identified and their deficiency symptoms discovered. It has been employed for the commercial production of crops like tomato, seedless cucumber and lettuce. To achieve optimum growth of plants, the solution must be adequately aerated and concentration of mineral nutrients must be maintained constant. Number 2, criteria of essentiality of an element. The element must be absolutely necessary for supporting normal growth and reproduction. And in the absence of the element, the plants do not complete their life cycle. The requirement of the element must be specific and not replaceable by another element. That is, deficiency of any one element cannot be met by supplying some other element. The element must be directly involved in the metabolism of the plant. Essential elements. Only a few elements have been found to be absolutely essential for the plant growth and metabolism. They are classified into two broad categories, macronutrients and micronutrients. Macronutrients. Macronutrients are those elements which are generally present in large amounts in the plant tissues, that is, in excess of 10 mole kg of dry matter. Example, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium and magnesium. Micronutrients or trace elements are those elements which are needed in minute quantities, that is 0.1 mg per gram of dry matter, that is boron, copper, iron, manganese, molybdenum, zinc, nickel and chlorine. Some plants also require traces of cobalt, selenium and silicon. Essential elements can be classified into four categories based on their diverse function as given below. As components of bimolecular and hence structural elements of cells, example, carbon, hydrogen, oxygen and nitrogen. As components of energy related chemical compounds, example, phosphorus in ATP, magnesium in chlorophyll. As activator or inhibitor of enzymes, example, Mg++ is an activator of ribulose biphosphate carboxylase and phosphoenoliferate carboxylase. Zn++ is an activator of alcohol dehydrogenase. MO activates nitrogenase. Elements that maintain the osmotic potential of a cell, example, potassium is involved in the opening and closing of stomata. Chlorine maintains the anion-cation balance and the osmotic potential of the cell. Number 4, role of macroelements and their deficiency symptoms in plants. First, nitrogen. It is absorbed as NO3+, NO2+ and NH4+ ions. Function, it is a major constituent of amino acid, proteins, nucleic acids, vitamins, etc. Deficiency symptoms. Chlorosis observed first in older leaves, stunned growth in plants, purple coloration due to anthocyanin development in the shoot axis surface, dormancy of lateral buds, delayed flowering, wrinkling of cereal grains. Next, sulfur. It is obtained as sulfates 50/- ions. Function, it is the constituents of amino acids methionine and cysteine. It also forms part of ferredoxin and vitamins like thiamine, biotin and coenzymes A deficiency. Symptoms, chlorosis of younger leaves, stunned growth of plants, accumulation of anthocyanin, purple coloration. Next, phosphorus. It is absorbed by plants in the form of phosphate ions, either as H2PO4- or HPO4 3- functions. It is a constituent of nucleotides and nucleic acid. It is present in cell membranes as phospholipids. It is involved in phosphorylation reactions and energy metabolism as ATP. Deficiency symptoms. Deficiency causes purple or red spots on leaves. Leaves also become dark dull green. There is a delay in seed germination. Premature fall of leaves and flower buds also occurs. Next, potassium. Potassium is taken in as potassium ions K+. It is more abundant in meristematic tissues like buds, root tips and young leaves. Function: it determines the anion cation benefice in cells. It controls the opening and closing of stomata. It maintains turgidity osmotic balance in cell. It activates a number of enzymes. It is also involved in protein synthesis. Deficiency syndrome: intervenal chlorosis, scorched leaf tips, loss of apical dominance and a bushy appearance of plants, loss of cambial activity, shorter internodes, disintegration of plastids, increased rate of respiration. Next, magnesium. It is absorbed as Mg++ ions by the plants. Functions: it is a constituent of chlorophyll, forms the central atom of the porphyrin ring. It maintains the ribosome structure for protein synthesis. It activates the enzymes of phosphate metabolism in respiration and photosynthesis. It is also involved in the synthesis of DNA, RNA and activation of enzymes. Deficiency symptoms: intervenal chlorosis and necrosis first occurs in of older leaves, premature leaf fall. Next, calcium. Calcium is obtained as calcium ions cat from the soil. Functions: it is necessary for selective permeability of cell membranes. It occurs as calcium pectate in the middle lamella of cell wall, hence is necessary for cell enlargement. It is used in the formation of mitotic spindles during cell division, hence is required in the meristematic and differentiating tissues, root apex and shoot apex. It activates certain enzymes in the metabolism. Deficiency symptoms: stunt growth, stunt growth, necrosis of meristematic regions. Number 5th, role of macronutrients and their deficiency symptoms. Iron is absorbed mainly in the form of ferric ions, Fe3+. Functions: it is an important constituent of cytochromes and ferridoxin, involved in the electron transport. It activates catalyze. It is involved in the synthesis of chlorophyll. Deficiency symptoms: chlorosis start with the intravenous regions and result in chlorosis of the complete leaf. Copper: it is absorbed as cupric ion Cu++. It functions: it is associated with certain enzymes involved in the redox reaction. It is essential for the overall metabolism. Deficiency symptoms: necrosis starts from the tip of young leaves and extend downwards. In fruit trees, it causes dieback of shoots, of shoot meristem. Leaves fall off and barks become rough and splits exuding gummy substances. Boron: it is absorbed as BO3 3- or B4O7 2-. Functions: it is required for carbohydrate translocation as sugar borate. It is necessary for uptake and utilization of calcium ions. It is needed for pollen germination. It is involved in cell elongation and cell differentiation. Deficiency symptoms: death of root and shoot tips, abscission of flowers, loss of apical dominance, absence of root nodules in leguminous plants, small sized fruits, stunned roots. Zinc: it is obtained by plants as Zn++ ions. Functions: it is necessary for the synthesis of auxins. It activates many enzymes, especially carboxylases. Deficiency symptom: malformed little leaves, intervenal chlorosis, stunned growth. Next, magnesium: it is obtained by plants as Mn++ ions. Functions: it is very necessary for photolysis of water in photosynthesis. It activates many enzymes involved in photosynthesis, nitrogen metabolism and respiration. Deficiency symptoms: chlorosis, gray spots on leaves. Molybdenum: plants obtain molybdenum as molybdate ions, MOO2O-. Function: it is a cofactor for enzyme nitrate reductase. It is also necessary for nitrogenase. Deficiency symptoms: intervenal chlorosis, whip-like disease in cauliflower, necrosis first observed in older leaves. Chlorine: it is taken in the form of chloride ions. Functions: along with Na+ and K+ ions, it determines the anion cation balance in cells and determines the solute concentration. It is necessary for photolysis of water in photosynthesis. It may be required for cell division in leaves and roots. Deficiency symptoms: wilt leaves, stunted root growth, reduced fruit formation. Deficiency symptom. An element is said to be deficient if it is present below its critical concentration. Since each element has one or more specific structural or functional role in plants, in the absence of any particular element, the plant shows certain morphological observable changes. They are indicative deficiency of a certain element and are called as deficiency symptoms. The common deficiency symptom include chlorosis. It is a loss of chlorophyll leading to yellowing of leaves. Deficiency of elements nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc and molybdenum cause chlorosis. Necrosis. It refers to the death of tissue, especially of leaves. Example, deficiency of elements like calcium, magnesium, copper, potassium. Inhibition of cell division. It is caused by deficiency of potassium, nitrogen and molybdenum. Delay in flowering. Deficiency of elements like nitrogen, sulfur and molybdenum causes delay in flowering. The deficiency symptoms tend to appear first in the young tissues in the case of elements that are relatively immobile and are not transported out of the mature organs. Example, sulfur and calcium, which form structural components. The deficiency symptoms tend to appear first in older tissue. If the elements are actively mobile, they are withdrawn from the senescing organs and transported to young tissues. Example, nitrogen, potassium and magnesium. Next, toxicity of micronutrients. An element or mineral ions is considered toxic if it reduces the dry weight of plant tissues by about 10%. The toxicity symptoms are difficult to identify. Toxicity levels of an element varies with the plant species. Excess of an element may inhibit the uptake of another element. Example, manganese toxicity produces brown spots surrounded by chlorotic veins. Manganese competes with iron and magnesium for uptake and with magnesium for binding with enzymes. Manganese also inhibits calcium translocation in shoot apex. As a result, excess of manganese may induce deficiency of iron, magnesium and calcium. Next, mechanism of absorption of element. The process of absorption occurs in two distinct phases. In the first phase, there is a rapid uptake of ions into the free space or outer space of cell called the apoplast. It is a passive process and takes place through non-channels. In the second phase, the ions are taken slowly into the inner space, the symplast of the cell. This is an active process and requires the expenditure of metabolic energy. Next, translocation of solutes. Mineral salts are transported through the xylem along with the ascending stream of water, ascent of sap, which is pulled by the transpiration pull. Next, metabolism of nitrogen. Nitrogen cycle. Nitrogen is a limiting nutrient for both natural and agricultural ecosystem. It exists as two nitrogen atoms held together by strong triple covalent bonds. N is equal to N. Nitrogen cycle involves the following steps: nitrogen fixation, ammonification, nitrification and denitrification. Nitrogen fixation. The process of conversion of nitrogen into ammonia and or other nitrogen compounds is known as nitrogen fixation. In nature, lighting and ultraviolet radiation provide energy to convert nitrogen into nitrogen compound like N2O, NO and NO2. Atmosphere also gets some amount of nitrogen oxide from forest fires, automobile exhaust, industrial combustion and power generating stations. Ammonification. The process by which organic nitrogenous compounds are decomposed to produce ammonia is known as ammonification. Some ammonia volatilizes and re-enters the atmosphere. Some of the ammonia is converted into nitrites and nitrates. Nitrification. Nitrification is a process of converting ammonia first into nitrate and then into nitrate. 2NH3 plus 3O2 gives rise to 2NO2 plus 2H plus 2H2O. 2NO2 plus O2 gives rise to 2NO3. These processes are carried out by soil bacteria that are chemototropes. Ammonia is oxidized into nitrates by nitrosomonas and nitrococcus. Nitrate is oxidized to nitrate by nitrobacter. The nitrates are absorbed by the plants and reduced to nitrites. The nitrites are transported to the leaves and reduced to ammonia, which form the amino group of amino acids. Denitrification. It is a process of conversion or reduction of the nitrate into free nitrogen. It is carried out by bacteria like Pseudomonas and Theobacillus. Biological nitrogen fixation. Though nitrogen is abundant in the atmosphere, only a few living organisms, especially certain prokaryotes, are capable of using it. The process by which living organisms convert the free atmospheric nitrogen into ammonia is called biological nitrogen fixation. The nitrogen fixing microorganisms may be free living or symbionts. Free-living nitrogen fixers includes Azotobacter, Rhodospirillum, Clostridium, and cyanobacteria like Nostoc and Anabinia. Symbiotic nitrogen fixer include Rhizobium in the root nodules of legumes, Frankia in non-legumes like Alnus and Casuarina, cyanobacteria like Nostoc and Anabia, cycas roots, Anthoceros, Azolla, etc. Symbiotic nitrogen fixation takes place in the root nodules of legumes. During the process, a nitrogen molecule is reduced by the addition of hydrogen atoms into two molecules of ammonia catalyzed by the enzymes nitrogenase. Biological nitrogen fixation requires the following three biochemical components: a reducing agent to transfer the hydrogen atoms to dinitrogen, ATP to provide energy, the enzyme system nitrogenase, MOFE protein, and ligamoglobin. The ligamoglobin is a pink color pigment similar to hemoglobin of vertebrates and function as an oxygen scavenger and protects the nitrogenase from oxygen. Here is the diagram for that. Next, formation of root nodules in legumes. When the root hair of the leguminous plants come in contact with rhizobium, it becomes curled or deformed due to chemical secreted by the bacterium. The rhizobia enter these deformed root hairs and proliferate with the root hairs. Root nodule formation in the leguminous plants. The plant responded by forming an infection thread that grows inwards to deliver the bacteria to the tissues. It is believed that the cytokinin produced by the bacteria and the auxin produced by the plant cells stimulate cell division and enlargement to form a nodule. The nodule establishes contact with the vascular tissues of the host for absorption of nutrients. The formation of root nodules and nitrogen fixation occur under the control of Nod genus of legumes and the Nod Nif and Fix genus of bacteria. Next, synthesis of amino acids. The ammonia formed by nitrogen fixation is used for the synthesis of amino acids. There are two processes by which amino acids are synthesized. One, reduction amination. In this process, ammonia reacts with alpha ketoglutaric acids and forms glutamic acids, a ketoglutaric acid plus NH3 plus NADPH, glutamic acid plus H2O plus NADP. The reaction is catalyzed by glutamate dehydrogenase. Next, transamination. In this process, the amino group is transferred from one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid which transfers its NH2 group to form 17 other amino acids. Here is a formula for that. Next is amides. Amides are formed by the replacement of hydroxyl ions of the amino acids by NH-2 radical. Asparagine and glutamine are the two major amides found in plants. They are formed aspartic acid and glutamic acid in the presence of enzymes asparagine synthetase and glutamine synthase respectively. Amides have more nitrogen than amino acids. So this is all about your notes. If you want the notes of more lessons, check the playlist of my channel. Thank you.